Okay, so we've seen how these high potential electrons from NADH get into the electron transport chain through complex 1. Now we've got to get those electrons from that reduced FAD, that FADH2 coming out of the succinate dehydrogenase reaction. Now we got to get those into the electron transport chain, right? Okay, this is of course the business end of complex 2. And then once we get through complex 2, we will have those two bifurcation points, the input from complex 1 for NADH, the input from complex 2 for FADH2, and they both converge on that coenzyme Q pool, which is then going to transfer its electrons to complex 3. So that's what we're going to take a look at here in this video. Okay, so let's talk a little bit again, like I said, about complex 2. Like I just mentioned, we've seen this process of bringing electrons in from NADH through complex 1. We're now, like I said, we're going to move on to bringing those electrons in from FADH2, that succinate dehydrogenase reaction, into the electron transport chain through complex 2. And this enzyme, or this complex 2, you've already seen before, right? This is succinate dehydrogenase. Complex 2 is succinate dehydrogenase by another name, right? As I mentioned before, when we were talking about TCA cycle, I mentioned there was one enzyme, only one, of all those enzymes, roughly 10 enzymes of the TCA cycle, that was a membrane associated protein. Specifically, it's an integral membrane protein. And now, after Dr. Carves listens, you know exactly what that means. Succinate dehydrogenase is an integral part of both the TCA cycle and the electron transport chain. It's the same protein, but one end of this molecule, this piece of it that is sort of sticking out of the mitochondrial matrix, is running this succinate dehydrogenase reaction, and it directly feeds those electrons straight into the electron transport process. So let's take a look at that, right? Okay, so once upon a time, I used to have to talk about this based upon an analog of an E. coli protein that was a quinol fumarate reductase. It was a structural analog, but it was, you know, a little bit different, uses a different electron carrier, whatever. But about 15 years ago or so, the structure of the bacterial, an X-ray structure, high resolution, 2.6 angstrom structure of the bacterial succinate dehydrogenase complex, or complex 2, was solved, right? And this is the structure of it, right? And so it's kind of neatly illustrates what's going on here. So complex 2, or succinate dehydrogenase, is a relatively large complex, about 140 kilodaltons, 4 subunits in each piece. And then there are 3 of these pieces, right? 4 tetramers, I guess you could say, arranged in a trimer. And you can sort of see a view of it here. So here's the membrane. Here's that mitochondrial intermembrane, that gray box. Here's one tetramer. Here's another one here. And there's another one back here behind that are arranged in sort of a trimer that are embedded with the membrane spanning domains down here in the mitochondrial intermembrane, right? Like I said, there are 4 subunits. One of the main subunits up here is what we would call that succinate dehydrogenase subunit. And this is where the business end of the reaction we've been talking about up to this point. That's where succinate would bind and be turned into fumarate, right? By transfer of those electrons to that covalently bound FAD molecule right there, right? Those electrons are then going to move down a iron sulfur chain, right? There are 3 iron sulfur clusters in here, right? 1, 2 iron, 2 sulfur, and 2, 4 iron, 4 sulfur clusters. And you can see, just like we saw when we talked about complex 1, we're sort of making a little iron sulfur cluster wire heading right down towards the membrane, right? And here, right at the edge of the membrane, sort of where the ectodomain, soluble domain of this protein now begins to make contact with the membrane down here, we see right here ubiquinol, right? That coenzyme Q molecule sitting right down there, very close to that last iron sulfur cluster. Now it turns out in these family of proteins, there's an interesting wrinkle here, which is that these proteins also have a bound B-type heme. Right? And when I say a B-type heme, we're going to see them again here in a little bit. We're talking about the same kind of iron protoporphyrin that we saw when we talked about hemoglobin, right? And the interesting thing is this heme doesn't seem to be involved in electron transport directly. It's not in the electron transport chain, right? So what happens when the succinate dehydrogenase subunit actually runs the oxidation reaction, sucks those electrons into the FADH2 here, and this is indicated in this slide as oxaloacetate. The structure was solved. The molecule here really is oxaloacetate. It's an inhibitor. Oxaloacetate is a competitive inhibitor of this enzyme, and it just co-purified with it, but it marks the active site. Those electrons go from FADH2 into this 2 iron 2 sulfur cluster, into this 4 iron 4 sulfur cluster. Sorry, then the last cluster is a 3 iron 4 sulfur cluster here, and there's the ubiquinol. And you can see in this structure, it's really pretty neat. There's a cardiolipin molecule bound, right, which is an important component of this intermitochondrial membrane, right? And there's the heme, this B-type heme that's off to the side. It's not in the direct electron transport chain at all. You can mutate the residues and eliminate that heme, and the electron transport will still work. You'll still reduce ubiquinone to hydroquinone. So it's an interesting question why it's there. I'm not sure anybody really knows. The speculation is that this heme is either structurally important or it's necessary for modulating the efficiency of electron transport into the ubiquinol to reduce the formation of reactive oxygen species in some way. But it's interesting that it's intimately snuggled up next to the ubiquinol, but not immediately necessary for electron transport, right? Now, similar theme we saw with complex 1. Notice the gaps, right, between the FAD and the 2 iron. Between each of these iron, we got 16 angstrom, 12 angstrom, 12 angstrom or so, 11 angstrom gaps. So again, we're sort of quantum tunneling the electrons across these gaps as they flow down to form dihydroquinone, right? Okay, that's more or less our input now. We've had complex 1 making dihydroquinone. We've now had complex 2 of succinate dehydrogenase making dihydroquinone reduced ubiquinone, right? So now what we've got to do is get the electrons out of that reduced ubiquinone pool, that dihydroquinone pool, into the next complex, right? And that is going to ultimately be the job of this next complex, complex 3. Now, this complex is an even larger complex. It's about 250 kilodaltons of molecular mass. Complex 3 is actually going to do something a little funky for us here, right? We're going to see that we've got an interesting problem to solve here, but this is a big complex, about 11 subunits. And it now is going to start using, we saw that complex 2 had a B-type heme in it, but it's not directly in the electron transport process. We're now going to start seeing hemes used for direct electron transfer, right? We're actually going to modulate the oxidation state of that iron that's bound in the middle of that tetrapyrrole, right? And complex 3 comes with 2 different classes of heme, a C-type heme, I'll show you what that it looks like in a minute, and a couple of B-type hemes, right? It's also got a really unusual iron sulfur cluster. All the iron sulfur clusters we've seen up to this point have been either the irons are coordinated with cysteines and or inorganic sulfur. In this so-called Riesk iron sulfur cluster, it's a 2 iron, 2 inorganic sulfur cluster. 2 of the coordination positions on one of the irons are cysteines, 2 on the other are nitrogens of a pair of histidines. Right? So it's very unusual in terms of its design. And the Riesk iron sulfur cluster is up here at the top of the complex, right? So if you're looking at this complex the way you've been thinking about it, down here is the matrix of the mitochondria. Out here, this is the intermembrane space, right? This is our intermitochondrial membrane. So up here is the intermembrane space, right? So outside relative to the matrix. And up near that top is that Riesk iron sulfur cluster, right? And this so-called C-type heme, right? Those are going to become important, right? Down here, sort of embedded in the sort of transmembrane region of complex 3 are these 2 other B-type hemes. And you're looking at that, well, and they're important for electron transfer, but you're thinking, why would they be down there? That's a little weird, right? Because ultimately, if you guys remember from learning this maybe as undergraduates, the electrons are going to flow into this small protein we're going to talk about here in a minute, again, called cytochrome C. It's up here. So what's going on down? Why would you have electron transport going down in here? Okay, so just to introduce this to you guys, remember those B-type hemes that we're talking about here. This again is basically iron protoporphyrin 9, basically the same thing as what we saw when we talked about hemoglobin. That C-type heme, however, has the ring substituents are a little bit different. In particular, two of those sort of exocyclic, the vinyl groups that are attached to the tetraperol are actually covalently connected through thioether bonds to the protein itself. So the C-type, B-type heme is bound really tightly. It's not coming on and off. But the C-type heme is actually, or the sort of C-type heme, also known as a cytochrome, right? These are B-type cytochromes, C-type cytochromes. The C-type cytochrome is covalently attached to the protein. Okay, what's with this? You know, you can already tell something weird's going on here. We've got this electron transport pathway, and the sort of idealized diagram here seems to bifurcate. What's going on? All right, well, if you think about this, up to this point, we've been transferring 2 electrons, right, and a proton, right, or 2 electrons and 2 protons in the case of FADH2, right? And now what we need to do, if you just remember the structure of those cytochromes we just talked about, that B-type or a C-type cytochrome, we've got 1 metal. And we can only change its oxidation state by 1 electron equivalent, iron 2 to iron 3, all right? So complex 3 essentially is a single electron transfer system. But the electron carriers that we're putting in NADH and FADH2 are 2 electron carriers. So somehow we've got to build an adapter between a 2 electron transport system and a single electron transport system. And the secret to this has to do with this sort of semiquinone radical form that coenzyme Q can pass through, and the nature of this bifurcated electron transport process, right? So again, you'll remember that coenzyme Q can form 2 different or sort of stepwise oxidation reductions, right? So we have the ubiquinone, that diaromatic diketone, take up 1 electron, 1 proton to form this ubisimiquinone, or radical form, take up another proton and electron, and then get the fully reduced sort of alcohol, dialcohol version, or dihydroquinone. Okay, so we're going to use that property combined with structure of complex 3 to build this 2 electron to 1 electron transfer process. And this is sometimes known as the Q cycle. All right, so here's how it works. So remember again, we've got that hydroquinone, so the dihydroquinone pool, fully reduced coenzyme Q. We've been feeding it from complex 1, complex 2. It's dissolved in the membrane, and they slide right on into that first binding site in complex 3. That's up here, right, sort of at the top of the transmembrane domain, up near the towards the surface of the membrane, as it comes up to the intermembrane space, right? Sort of the outside, if you want to think about it that way. And in this first binding site, there's a bifurcated electron transfer, right? One of the electrons moves through that Riesz iron sulfur cluster and then into that C-type heme, that C-type cytochrome right at the top of the complex. The other electron moves down into that through those B-type hemes, right, down into a second binding site where there is a binding site for an oxidized coenzyme Q, a ubiquinone, right, fully oxidized. And it takes up 1 of those electrons and forms that radical, or a ubisimiquinone, right? Now, you'll notice something, right? Remember from we talked about before, and I didn't point this out as explicitly of complex 2, but the same thing's happening in complex 2. In the case of complex 1, it's both pumping protons, right? But it's also taking up some chemical protons when we make this reduced dihydroquinone Q. Complex 2 doesn't pump protons, but it still takes up a couple of protons in the case of chemical protons for the coenzyme Q pool. So if you look here, we bring these guys in and we now bifurcate these electrons. One goes down into the B-type site to form that ubisimiquinone. The other flows up into that C-type cytochrome through the Riesz center. And at the same time, two of two protons are now ejected to the side of the inner membrane space, the outside of the inner mitochondrial membrane, as we reoxidize that coenzyme Q that was in that first binding site, right? So you can think about this as almost like a redox cycle, like I talked about before, where these guys were chemical protons were taken up sort of on that inner surface, right? We saw in both the case of complex 1 and 2, where we're forming that reduced coenzyme Q pool, that dihydroquinone. But now we oxidize on the sort of on the other side, the upper face of the membrane, and the structure of the protein is designed to eject those protons as we pull the electrons out into the inner membrane space, right? So we get some pump some chemical protons as we go through the first half of the Q cycle. All right, so now what? Okay, well, we got one electron out, and we got this radical hanging out down here. Yeah, that's a problem. Okay, well, what happens is this oxidized Q from this first binding site is released back into the mitochondrial membrane as the ubiquinone, the fully oxidized aromatic diketone. The semiquinone version one that was in the second binding site, it stays there. And what happens now is a second molecule of dihydroquinone binds to that first site, and we repeat the process, right? We now peel one electron off back to the risk iron sulfur cluster, and then into that C-type cytochrome, which then gets transferred to a second molecule of cytochrome C. I think I forgot to mention that first electron goes into that small transport protein that's hanging out on the outer surface of the mitochondrial membrane, inner mitochondrial membrane, cytochrome C. We're going to talk about that in just a minute. And once that's reduced, it goes away, it diffuses away. The second one comes in on the second half of the Q cycle. You transfer another electron there. That's now one equivalent of a dihydroquinone, right? We put one electron here, pulled another one here. That's an equivalent, two equivalent, but we did it in a single electron step, one, then the other. That other electron then, because again, we're going to split, comes down back into that second binding site through those B-type cytochromes, and that reduces now that semiquinone that was hanging out down there, and now makes dihydroquinone, which is now released back into the Q pool, right? So the whole point of the Q cycle is to allow this 2-electron dihydroquinone carrier to adapt essentially to a single electron carrier, which is cytochrome C, right, which we're going to see here in just a second, which is a single heme bound with a single iron that it can toggle. And so by splitting it this way, and by doing 2 steps of electron transfer, you can now adapt this 2-electron carrier to a single electron carrier, right? Okay. So the second part of this process, or the remaining part of this process, is we now need to get those electrons into cytochrome C, right? And that's our small electron carrier. Cytochrome C is a roughly 100 kilodalton amino acid, about a 12 kilodalton membrane-associated protein. It's not a transmembrane protein itself, but it has a very strong affinity for hanging out, sort of surfing along the outer surface of the inner mitochondrial membrane, right? It doesn't freely diffuse out in the inner membrane space very much. It really wants to hang out here along the inner mitochondrial membrane. It sort of diffuses along it, looking for complex 3s that have a reduced C-type cytochrome at the top of the complex, right? So the cytochrome in cytochrome C, again, is a C-type cytochrome like we just looked at. It's covalently bound within this little 100 kilodalton protein, but it's not fully encased, right? So if you look at this, there's the C-type cytochrome down in this little 12 kD protein, cytochrome C, and you'll notice that it's mostly sort of encased in the protein, but not totally. There's one little edge of that cytochrome that's sticking out right at the surface, right? And that little edge becomes really important because when you pull the electron, so again, the ubiquinone, dihydroubiquinone comes in, with that primary site, comes in and splits those electrons, right? Here's that risk iron sulfur, there's the C-type hydrochrome, there's the B-type cytochromes, the second binding ubiquinone binding site is down here. The top pathway goes up, and when this C-type cytochrome at the top, right, sticking out of the complex, it sticks out in the in-membrane space, that gets reduced. This recruits a cytochrome C that then binds, and you'll notice something. Look at that C-type cytochrome that's in complex 3 and the C-type cytochrome that's in the protein cytochrome C, and you'll notice something, right? they get really close to each other, right? They are almost in van der Waals contact. So it's essentially what happens is as soon as that C-type cytochrome in complex 3 gets reduced, and within the window of time it then takes for an oxidized cytochrome C to then bind, the electron transfer happens really fast. It's like on the microsecond time scale, because these complexes get, these cytochromes, these electron transport centers get so close to each other that electron transfer here is super fast, right? Okay, so with that, we will then have taken those electrons out of complex 3, put them into this little diffusible carrier, cytochrome C, and now we're going to move them into the final stage of this electron transport process, where we're going to ultimately make water and dump those electrons off, right? Okay, so just as a quick review again, cytochrome C, complex 3 has pumped some protons, they're chemical protons, right? Complex 1 pumped protons, both by taking up protons out of the matrix as we formed reduced coenzyme Q. And it also, remember those 4 proton pumps, it pumped protons. So it has structural protons it's pumping, and it's taking up chemical protons. Complex 2 doesn't directly pump any protons. There's a little bit of an uptake as a result of forming dihydroquinone. And complex 3 is now moving protons through sort of a redox cycle type release of those protons as these ubiquinones get oxidized on the cytoplasmic face and those protons come out, right? Okay, now we'll move on in the next video to the final step of this process, which is transfer of the electrons from cytochrome C to complex 4.